So now we're going to have an open discussion and question and answer period. So could all the speakers come back up to the front, please? Thank you. And I have questions I could pose, but I will wait. I will let everybody here ask questions first. They can be questions to individual speakers about their talks or general questions to the entire panel of speakers. So,
Shelly, you're joining the mic?
Yes, please.
Okay. For the web, yeah. Okay. Jenna's going to get it.
Okay. Yeah, there.
Shelly, I'm intrigued by the variability study. And I'm wondering if you have a sense of how the variability over short term compares to the point-to-point variability if you were sampling multiple locations within the home.
Yeah, unfortunately, we only sampled in one location, but we did find the variability from day to day was just as high as from home to home and from time farther away. So we only had one location though.
I also had a question sparked by what Shelly talked about, but I think all the speakers might have thoughts about this. So you described that fungal variation in a geographic context was more resilient or less than bacterial variation. And you also described that at a single location you had more variation for bacteria than fungi. On average, across all your different study systems, are you seeing this general pattern that fungi have less variation either across space and time than bacteria? Or even you can talk about different groups of genes or organisms, not just bacteria and fungi.
We are seeing this geographical impact on the fungi, and we don't yet have we don't have the fungi data for our home study. We didn't have enough sample for that, so I can't really comment on that particular issue. But we're seeing that the indoor environment, even in other studies, the studies in Bay Area show that the fungal communities are really associated with outdoor environments and the bacteria with who's inside.
Yeah, I mean, in some of our systems, fungi are more abundant, at least in the soil as well, but we're finding in the air there are more fungi, but a lot of them, at least at first, we missed because they're in spore form, so that might be why they're carried a little bit easier, but I mean, it makes sense from Shelly's results that maybe a lot of the bacteria are sort of moving around more through organisms, since microbiomes and gut or human associated are more bacteria, and maybe in some some soils anyway, fungi dominate. So.
I want to thank all three speakers. These have been really terrific talks. And I have a follow-up question in regards to the variability indoors and outdoors. Do you think it's a contradiction or a function of sampling approaches that you found that there was a lot of variability over time of indoor as well as outdoor bacteria, and yet the bacteria indoors were predominantly determined by the people indoors who weren't varying all that much, I would assume? Is that question clear, or?
Can you repeat the question?
Yeah. One would think that if people if the people in the house aren't changing over time, that the indoor bacterial communities would be stable, relatively stable. And yet, you're reporting that they're varying a lot. Both may be true, but I'm trying to connect the dots.
Yeah, I think because only a very small part of the community is associated with indoors. So for fungi, it was 1.1% of the taxa were predominantly indoors, and for bacteria, it was 1.6%. So it's a very small fraction of the community, and so that the variability can still be very, very high, but when we look at what's indoors, that little fraction is very stable. Does that make sense? Yeah, I hope so.
Over here.
I have a question for Sarah about vocabulary. You used the word resilience and you used the term tolerance. And I'm wondering, do you mean what some people call stress tolerant, could you define those terms a little bit more precisely?
Yeah, that's a good question. I think that sort of a persistent problem, even in the ecological literature, with how people define those, and even just the framework for describing these perturbation response. So generally, resilience is the idea that a system can be perturbed but then return to the original state, and resistance is that it's not really changes a lot in the first place despite the perturbation. So that's the distinction between the two, but the question comes in as sort of what the response variable is. If a lot of times when I was talking about the community actually changing, so not necessarily being resilient, resistant to the perturbation, but maybe it's a biomass or something else because sort of it's adapting that does either bounce back or is resistant just because they're acquiring sort of more tolerant species. So yeah, there's a lot of vocabulary associated with this, and stress, sometimes people define as a more short-term response or short-term perturbation that doesn't necessarily cause like widespread death or something, and disturbance is more sort of a longer-term thing that causes some species to die. So yeah, there's a lot of definitions associated.
Because we have so many communities coming together as we study the built environment, we find that vocabulary can be really important, that people from different subdisciplines can be using the same word to have somewhat different meanings. And I think as we go forward, it's important for us to say what we mean, like perturbation or stress or whatever. Okay, thank you.
Great. I think the next question is in the back, and then this gentleman in the middle here. Okay.
Sarah, I have a question for you. In your grass experiment, how did you group the taxa into tolerant or opportunistic? Was that done a priori based on natural history, or?
No, it was based on clustering of their responses over time. So we basically, I didn't go into it in that much detail, but sort of if a certain taxa was sort of low abundance at the beginning of this, so the disturbance itself is sort of a time series, so like 3 points on multiple pulses. So if taxa were low at the beginning or sort of average at the beginning and then ended up high in abundance after these repeated disturbance, we considered them tolerant. So it's really, I mean, operationally defined. We just call them those names, sort of goes back to definitions where in the case of this study, they withstood that disturbance and were high abundance at the end, whereas the sensitive species we labeled as they decrease. So.
This gentleman right here.
Thank you very much for a good presentation. So there are two questions, one directly to Sarah and the other one for all three members. So Sarah, again, we are talking about tolerance here. So it is my comment that, I am really surprised when you said fungi are more tolerant than bacteria, but even if you go to the Atacama Desert, what we have seen are mostly the bacterial members who are tolerant to that soil, which is highly desiccated. And maybe your agricultural soil may have some sort of moisture compared to the highly desiccated Atacama Desert soils. So in our opinion and other wealth of research saying that bacterial spores and Deinococcus kind of things are more resistant to those tolerance. This is just a comment. I'm not expecting any answer back because we pounded on tolerance so much here. But my general question for all three of you are, how do you define that what you have seen in these systems are viable? In other words, how do you differentiate the dead signatures? Because most of them are based on genome, except Janet, who had done some transcriptomics analysis.
Yeah, I think I can take that one. So we worry, well, this is a very important consideration for permafrost, where we expect to see a lot of dormant cells. And also, to your first point, those systems are highly bacterial dominated. And we have a lot of actinobacteria that are tolerant to that sub-zero temperature condition, and they're known to be able to form this dormant sporulating state. But because we had the RNA data and the protein data, at least that is a sign that there is activity. And then ultimately you need to try to isolate or do some kind of activity measurement. We were doing gas flux measurements to see that there really was activity, and doing some other kinds of biogeochemical activity measurements to add that layer of validation.
So I agree that's a worthwhile, it's something that is think about a lot in soils, as most of the taxa are dormant. There's multiple ways to do that. I've used the nucleotide labeling that is labeling the new DNA that gets replicated, and then RNA, in looking at RNA to DNA ratios. There's also isotope labeling of different compounds, stable isotope probing, where you're looking at which microbes are actually incorporating this compound into their biomass, and you can separate them out that way. So it's sort of just a matter of what the question is, what you're using. I think the challenge with that is that even with RNA, it's almost like there's not a good in-between, where with RNA you're really getting a snapshot of what's happening right then. And if we're talking about just variation in environments over time, that day versus that night might see different active community that's not really representative of that long-term environment. But DNA might be representative of many, many years. So, but yeah, I think that's something that we should do.
So Bob, can I ask a question from Vivian?
Yes.
So Vivian Lofness is one of our committee members. She's participating remotely, and she had a question for Janet. Is more research needed on early life exposure signatures and their likelihood to carry through to later environments, in your experience?
Yes.
Okay, well then I'll ask another one. To Sarah, she asks, if you want to keep microbe-rich soils, should you water your gardens? Should you try and condense moisture out of the air? I think she's trying to get at an extrapolation of what you've seen.
Yeah, I think that you should water your gardens in general. Microbes are dependent on water, and in general trends, wetter environments have higher microbial biomass up to a certain point, where other things are limiting, sort of just like plants. So I think a lot of the systems I work on are already low biomass, so they don't necessarily show the same response to, say, watering as a wet environment. So does that answer your question?
Over here is next.
My question is regarding bacteria. So at the core of the dispersion and persistence of bacteria is their ability to sporulate. So what do we know about sporulation of bacteria? What percentage of all these groups that we find are able to sporulate some way or another?
So percentage, we know that some groups have a lot of spore-forming members. So the bacillus are the paradigm for being able to form spores. And actinobacteria that I just mentioned, they also, sometimes some of those form spores, but some of them just kind of round up into a dormant state and harden their their outer surface, they become more resilient to stress without forming a real spore. And so you have this gradation of ways of coping and becoming dormant.
I think you were next.
So this is first for Sarah, but I'd like everyone's opinion, please. You were talking about dispersal and that the rain can carry microbes, both fungi and bacteria. So in relation to buildings that leak, do you think that water leaks into a building from rain would carry these microbes in with it? And could that change over time? The types of microbes that are in the rain?
I mean, definitely the types that are in rain changes over time. We really tried to get a representation of what's in the rain coming from the air or the sky. But I know of other studies that I think that they were looking more at like stormwater drainage that really show that changes a lot based on once it hit the surface. And even when, we would kind of disregard a sample if there was like a branch in it or something, or, once it hits a surface, that there's so many more bacteria and fungi on those surfaces that it really changes the signature of that and increases the biomass too. So yeah, I think it would change it. And most rain that's going into environments that, indoor environments have hit something before they get there.
Question over here.
Hi, this question is for Janet. I wanted to get more of your sort of thoughts on the integrative omics concept that you were talking about. How orthogonal is each sort of analysis procedure versus interdependent on sort of the previous? So for the metagenomic analysis, from a genomic perspective, does that feed into the mass spec measurements? And do you sort of long-term envision sort of picking out in the future being able to identify sort of more important measurements that don't rely on all of them, or do you sort of see going forward a much more integrative approach?
Yeah, so to answer your last part first, I think we absolutely want to get to a stage where we can be more predictive instead of having to rely on these enormous data sets that have to be mined individually. So the hope is to have a model with a limited data set that you can predict what is occurring. We're not at that stage yet, and so the different kinds of omics data definitely do build upon each other. We're totally dependent on metagenomes that are well assembled for our metaproteomes, for example, because we use that as a database for our metaproteome searches. And similarly, for the metabolite data, we need to map that back to some kind of pathway. So we usually have a pathway, if you think of a KEGG pathway, and you can put the metabolites in there based on their predictions within a pathway, and then use that same map to put the proteins that would be also involved in those enzymatic reactions. And so it really is back and forth across the different data sets.
If I could indulge myself and follow up on this, it seems to me that you need both, because what you have with the genetic data is what you might think of as a potential community that's there that might become active if circumstances change. So the permafrost has just a few fascinating organisms that have iron metabolism, and it's amazing anything's happening at all probably. But if it melts, there may be a whole community sitting there, latent, ready to go, that got there thousands of years ago. And that's important to know as well. So I think there's value in both on the genetic side and characterizing the potential community, as well as characterizing the active community. I have another general point I'd like to ask all of you, which is, it has to do with the methodology of how we go about doing our studies. One thing I thought was fascinating, and what two of the speakers did, was they talked about deliberate experiments, where they kicked the world, they did a manipulation, and they watched what happened. You could imagine doing that in the built environment. You could take your college dorm and move boys to girls' rooms and girls to boys' room and say, how long does it take for that difference? Is there a sudden flush of an increase in diversity because you've mixed up the two types, and then it decays over a period of weeks to months to years? I think we should think about what kinds of deliberate experiments could be done in the built environment that would build on the omics tools, the ecological theory, and so forth. And I hope that's something we can have in our report.
If I could make a comment, I think it's also very important about the idea of figuring out the activity in a built environment, because personally, I care about what's in the built environment because people are there and I want them to be healthy and happy. And until I know whether the organisms there are, how they're impacting that, and whether they're actively participating and helping the participants be healthy and happy, then I can't say anything about how useful these communities are in the building.
Yes, back there.
I wanted to follow up a little bit on a question that Jessica asked earlier in terms of the difference between fungi and bacteria and the effect of humans on that. And this is related a little bit to what Sarah was talking about in terms of dispersal. I mean, it seems to me much more likely that geography is affecting bacteria indoors like it is affecting fungi, but that there's this really strong homogeneizing effect, or basically humans are a really important factor that's sort of overlaying this background of geographic variation in bacteria, and whether that's a consequence of the way we measure it and the way we study it, or whether that is just speaking to sort of the strength of the humans as vectors. But it seems like there's data that shows, or we would sort of expect, like from Sarah's data, there would be geographic variation in bacteria when you move 1,000 kilometers, right? And so we would expect to see that in your home, but we don't necessarily see it in this 2% of bacteria that are human associated. So, I mean, do you guys have a sense of sort of what is driving this disconnect, and like why we're not seeing geographic variation in bacteria in homes?
I mean, I think it sort of comes down to almost selection strength, or sort of, you could also think of it as a mass balance thing, where it's, think about how many bacteria are in homes that are transmitted by humans, how much that surface interacts with humans, maybe just overpowers sort of, I mean, there is geographic outdoor variation in bacteria, but maybe just the number of cells that humans are transmitting, and just sort of overpowers that variation, whereas there's just not as much of an imprint of humans on fungi.
Yeah, I was gonna look back at the other outdoor dust paper to see what the geographical distribution is from that paper, but I can't remember offhand.
Over here.
Yeah, very last one. My question is, how reproducible and quantitative for the water economics and photonomics technology right now?
It's technically highly reproducible between runs. So that reproducibility isn't really especially for proteomics. So for RNA, I would say it's less reproducible mainly because of extraction difficulties. But for the proteomics within technical runs, it's highly reproducible. The problem really is the dynamic range and the large number of unknowns that we have. That's the big problem with those technologies. It's we've been doing relative abundance, so it's quantitative but in a relative way with all of our assays. But we could do absolute quantitation. In that case, you would have to incorporate a standard or something. We are not doing that, but it's possible to do that.
Another question for Shelly. When you made this field environment diversity, so generally speaking, do a good job for the microbial community. But we did not collect much of the metadata, in the field environment, what came through the community. Like Sarah mentioned, the dispersal could be important selection. I guess the field environment, the dispersal could be more important than selection. But we don't have much data about introduction affect this community structure. So what kind of metadata we should measure in the building environment?
Well, yeah, we measured a ton of metadata in our studies, and yet very few of the parameters are associated with diversity and abundance, unfortunately. We saw some relationship with relative humidity and temperature in our 15-home study. In the 1,200 home study, the fungal communities are associated with whether there was a basement or not. Just a handful of parameters. And so that's why I didn't really mention too many of them in our studies. And just a comment on Rachel's question, in the PNAS paper, we do see the outdoor bacterial communities are associated with climate and soil. It's just the indoor ones, I think, as you mentioned, are swamped by the people, and so we don't see this geographical signal.
So Bob, I think we're going to do just one more.
Yes, one more question out there. Yes, okay, you're in the front.
The term reproducibility was just used, and this has been on my mind for quite some time. And I'd like perhaps each of you to define precisely what you mean by the term reproducibility.
Well, I was talking about technical reproducibility, but that's it. So that means if you do series of runs, technical runs, so you have within the machine, because it's a big.
Replication on the same sample.
Mm-hmm. It's replication on the same sample, so the technical runs with the machine. If you go, so that there, for the analysis that I was talking about, the technical reproducibility is very high. If you start going out into between sample reproducibility, or the degree of variation that you have between samples, then it starts to expand beyond that.
Yeah, it's just the same. I think of reproducibility as a methodological issue, where you're hoping for reproducibility among the same sample, but sometimes, depending on extraction bias or even machine bias, you don't get that, and you want to be confident in your data. But reproducibility, say over time, is just data to me. I mean, if the indoor microbiome changes over time, that's not a problem. It's more a methodological issue.
So let me just ask quick follow-ups. In the world that I come from, which is more general environmental engineering and science, reproducibility is usually based on split samples end-to-end. Have any studies been done on split sample end-to-end reproducibility that you're aware of?
You mean end-to-end, like through the whole process of analyzing it?
Yes. Well, no, that's even further. Even within the same lab, starting de novo from a sample split.
We haven't done that in our studies. We do multiple samples and then combine the sample, and typically we're struggling with enough biomass anyways, so we have not typically split a sample.
Yeah, I mean, we usually do some sort of test like that with a new technique. I think the problem in soils anyway is that you can't truly split a sample if there's spatial heterogeneity at a small scale. You can't sample the same spot in the soil twice. So we either solve that by splitting at a small scale, or kind of taking several samples and homogenizing to try to integrate across that. But you still can split a sample if you're going to mix it up and say, this is my biological sample unit, and then split it and do it the same way. So yeah, I mean, I think maybe it's not done enough, but we try to do that with a new process that we think there's some point in that pipeline that's going to be biased in some way.
Yeah, and I'll just add to that it's what you're saying is absolutely critical for development of a new technology. And so when we first started using proteomics, we did just that for new kinds of environmental samples, all the way from the extraction, all the way through the pipeline, to look at the reproducibility at all of the different steps. it's absolutely critical to do that. So,
One thing that I don't think people really test reproducibility in terms of even just 16S sequencing runs. There's some variability across runs. But I know we're trying to get going in our lab, just the idea of putting sort of a community standard in these runs, where it's sort of just a mock community of several species of known abundance, and then you can kind of check to see if we know the abundance of this mock community, we can kind of see how well the rest of it is represented on sort of unknown samples.
Yeah, so it's interesting. This question is related to Joan's question, which has to do, we use terms like reproducibility, resilience, robustness, and so forth. Are we all using the same term in the same way? It's important that we be clear about that in our report and science in general. And I'd like to thank the three speakers for really great presentations. Very stimulating. Thank you.